A couple weeks ago, I led my second workshop at Food Lab, and it was an absolute blast. Thanks to everybody who came out. I gave a lecture on fungi and how they work and why they're interesting, and then we prepared some of our own growing jars and inoculated them. I tried to record as much of the workshop as I could and have edited it slightly to compress things a bit. Several weeks ago, I live-streamed the first lecture and workshop I hosted, and people seemed to really enjoy it even though the video quality was terrible. So I'm going to be editing and uploading them this way as I didn't find the live stream really added to it. I'll be hosting another Intro to Bio workshop in the near future, and I'll re-record and upload a better version of that eventually. And of course, I'll be posting all of the dates for upcoming workshops once things have been officially scheduled. Let me know in the comments what you think about this new lecture series. I'll be hosting a lot of workshops soon, so if this is a format you guys enjoy, I'll release some of these more often. Now with that out of the way, let's get to the lecture. We started with media preparation because everything had to go into the autoclave to be sterilized, which takes a while. Here we're using the classic brown rice flour tech in a mix of 2 parts vermiculite, 1 part brown rice flour, and 1 part water. Everything is mixed and packed into jars, leaving about an inch of headspace. The lids of the jars previously had had 2 small holes drilled near the edges, which were sealed with silicone high-temp gasket sealant, and 1 larger hole in the middle for air. Once the jars were packed, a piece of filter paper was taped over the hole. Don't pack the media in tightly, make sure to leave it loose, and cover it with a thin layer of plain vermiculite on the top. Once the jars are sealed, they're given a tinfoil hat and then placed into the autoclave.
Yay!
Well, now that we've done a little bit of the hands-on stuff, I'd like to officially welcome you to the Fungi Workshop. Woo! Yay! But no, so I'm gonna try and cover as much about fungi as I can in however long it takes for the autoclave to heat up. Fungi are really, really fascinating. It's a whole fascinating area of the tree of life. It's also one of the least studied, because everyone's like, woo, plants! And they totally forget about fungi, even though fungi are, in my opinion, frankly, way more interesting. Because they can do weird things and, in the words of my plant professor, they're basically sessile animals. When I was actually learning about, all the different, kingdoms and groups of the natural world, in university, one of the things we had to learn was that we are in the group of epithsicons, which is a hard thing to say, but it's basically the whole thing that makes us special is that at some point in our life cycle we are a sperm with a tail facing backwards, which I realize is a pretty low bar as things go, because there's 3 things that fit into that category: all animals, all fungi, or most fungi or some fungi at the very least, and these weird single-celled things called epithsicons. So we're all in that weird group. But fungi specifically are particularly weird. They handle things very differently. Their life cycles can vary wildly based on the type of fungus that they are. And more to the point, the thing that if I say fungus, there's 2 things that generally come to mind, which are mushrooms and mold. The thing is, neither of these things are the most common. I mean, molds kind of, but like the mushrooms are the tiniest part of the organism. They're actually the reproductive bit and nothing else. The actual fungus itself is huge. The biggest organism on Earth is a fungus. It lives in this ash forest, I believe. And my professor was the one who actually figured out that every sample of fungus that they took from this forest of the same type of species was the same organism. They're like, why are all these genetic results like exactly the same, even though so the thing is like 3.5 kilometers wide. It's very big, but it's all interconnected. So fungi are mostly like this. They're mostly these little strands of stuff just spread out through some other thing, be it soil or a log or something else. So there's a bunch of different kinds of fungi. The ones we're most used to are these 2 main groups, but even in the groups you can see there's massive variation in the kinds of things that are here. So in the Ascomycota, you've got things like truffles and morel mushrooms, but you've also got like penicillium molds. So it really is a pretty wide spread. The other thing in here are baker's yeast. So in case you didn't know, yeast is also a type of fungus. They're kind of weird, and I omitted their life cycle, although I regret it now because it's actually kind of funny. But yeah, they're kind of interesting. But the things that we tend to picture of, like when I say like mushrooms, are the Basidium micata. They're the main like mushroom species, with the exception of morels. So these are the things that tend to grow on logs or in soil, and at some point in their life cycle they make a mushroom. So those are the most common. Those are the ones we're actually going to be looking at the most today. But just for the sake of it, all the other ones tend to be like diseases. So they'll either infect animals or plants or something else, and yeah, they don't really do much other than that. They're basically just disease. So yeah, mushrooms, everything else we find useful, and then disease is the easiest way to divvy that up. So, as I was saying, there's a lot more to a fungus than the bit that you see. So we're used to like this, which is a fairy ring. There's a fungus throughout the entirety of this underneath. So the thing is, most plants, we're used to the idea that you put a plant in the ground and it makes its little roots, and then the roots do all the hard work of collecting nutrients and stuff. They don't, actually. Most of what they do is they make friends with the fungus, and the fungus does all the work for them. So over there you can actually see what this looks like. So those are plant root cells, and the stripy bits are actually the individual little strands of fungus that grow into the plant, like into the cells, and form this symbiotic relationship. So the fungi will give the plant things like phosphorus, which plants have a very hard time getting enough of, and in exchange the fungus gets some sugars. And so it's this trade-off. But the interesting thing is, this doesn't just happen to one plant. All of the plants in the area are connected to this fungus, and potentially the same fungus. So what this ends up turning into is essentially like an underground internet of sorts, connecting all the different plants together. And they can use this to like communicate, or like restrict nutrient flow, or promote nutrient flow. So when like a mother plant puts down a bunch of seeds, if that plant detects that there's really good conditions, it'll pump more nutrients through the fungus into all the little baby plants to help that flow. Or if there's like like a parasite of some sort, something that's chewing on a plant, like the smell of cut grass is plants screaming. This is something is chewing on me. Oh God, make more bitter things so they don't chew on you. One of the other ways that they communicate is through mushrooms. Because it really is like an internet. It's truly fascinating stuff. The individual strands themselves are called hyphae, but the collection of all of this, the actual like whole mass of growing mushroom, is called mycelium. So the jars that we just prepared are going to get totally encased in this mycelium. So you're going to see them go basically totally white. So that's... Okay, yeah, so as I mentioned, the mushroom is just the reproductive bit. And also delicious, sometimes. But so the actual main life cycle, the whole point of a mushroom is to distribute spores. So spores are basically the mushroom equivalent of a seed. So the spores are produced in the mushroom, and once the mushroom is ready, underneath here is what's called gills. The gills are laced with all of these spore-producing cells. So at the right time, all the spores get released, not necessarily all at the same time, but they'll either slowly come out. Sometimes they are kind of pressurized, so when they go, they really go. But once the spores come out, they float away, or get carried on something, or are generally transported to another location. They land somewhere, and just like a plant, they spread out and they start growing. But what ends up happening is multiple different spores will each send out their little strands until they meet from another, like one spore's little strands meet up another spore's little strands, and they'll actually mate and form this new kind of fusion of tubules. It's really an odd process. A lot of like cells combining in weird ways that are not a thing you tend to think of when you're used to the ways that like animal cells work. But so they end up with these really interesting so you can see it over here. So if one type is red, one type is green, at some point they meet, and then after the meeting you get this weird mix of red and green. So then basically at that point they're just waiting. They're waiting for the ideal conditions. So generally after rain, this is why, especially in like the Pacific Northwest, all the mushroom hunters go out after it rained, because that's when all the mushrooms are like, ooh, perfect conditions, and so they all pop up. And in the span of a day, maybe two, you'll go from no mushrooms to everything covered in mushrooms. But that's the basic life cycle of the Basidiomycota. The Ascomycota, the things like yeast, they don't really do mushrooms. They tend to do, they still will do spores. Yeast specifically tend to do what's called budding. So basically the little yeast cell will grow a little baby cell off it until it's at the right size, and then they separate. So with Ascomycota, they tend to do mostly asexual reproduction, so it's things like budding. They can do sexual reproduction as well, and so the technical term for yeast sexual reproduction is called schmooing. I kid you not.
Making that up.
Yeah. So if you've ever heard me say, now make the thing schmoo, that's where that comes from. Yeah, the yeast end up very pointy, and then the pointy bits merge, and then they make little baby yeasts. So yeah, schmooing, there's your term for the day. So the weird thing with fungi is that as weird as they are, there is still stranger, but also they can form really, really interesting symbiotic relationships. So on the left here is Lichen. This particular species, I don't know what this particular species is, but at first glance you'd almost think it was a plant. And this is because it's not just a fungus. It's actually a symbiotic relationship between some type of photosynthetic thing, be it algae or other tiny little photosynthetic microbe, and at least one, usually two different species of fungus. And they do this really weird self-assembly thing where they end up making what is an entirely new species. Like, individual lichen are actually classed as their own species, even though they're a mixture of different organisms, which is a really strange concept. But then it gets into things like biocrusts, which is the same idea. It's just these weird collections of organisms that always exist together. So if you've ever had kombucha, the thing that grows at the top of the kombucha, it's called the SCOBY, which is literally symbiotic colony of bacteria and yeast. So they do this same thing, where it's some type of fungus, in this case yeast, and some type of bacteria that always come together. You can separate them, they really don't like it. And they grow way better together. And so there's a lot of this in nature. On the other hand, things that aren't a fungus, that are terribly named, slime mold. Slime mold, all of that is one cell, which is really weird. That's one of their defining characteristics. It's one giant goopy cell that's all strandy and strange. They're very strange, and even though they say mold in the name, they're not actually fungus. They're their own weird category of, we don't really know what to do with them, but they're definitely not a fungus. Because they don't do any of the usual fungusy things. Like, they'll live on wood, but they don't... Yeah, they're weird. They've got their own rule set, so they get put in their own bucket. Yeah, so as I was saying before, these are the main two that we're going to be looking at. So the Basidiomycota, you can see these are all proper mushrooms, right? So you got your reishi, enoki, the oysters, and shiitake. Whereas your Ascomycota are things like morels, but also the blue cheese mold. So the reason blue cheese is blue is because you make your cheese, and then once your cheese is ready, you take literally a spike and dip it in the spores of this type of fungus and stab it into the cheese, and then it spreads out and makes blue cheese. Same with yeast. But yeah, so basically the main role, or the most common role of most fungi, is they break things down. That's sort of their defining characteristic. So when lignin, the thing that the molecule that makes plants woody, was first, when it first happened. The first plants that could make it, there was, I believe, a 50 million year gap between plants figuring out how to make lignin and something else figuring out how to break it down. So there was just these massive piles of wood everywhere because nothing was eating it because they couldn't. And then the fungi eventually figured it out and they're like, ooh, well, there's kind of a lot of this now. But they do a lot of that kind of stuff. So the main fungus that we're going to be working with today are what are called white rot fungi. They eat dead wood. Some of them it will interact with the still alive trees. So there's this really interesting ecological niche where before we hunted them all, the bears would show up and they'd scratch the trees, which would damage the trees but let spores get in. So then all the different mushrooms would get into there, colonize the tree, which would inevitably kill the tree. But it was also how the mushrooms persisted. There's also now some research that those specific types of mushrooms, bees would actually drink the mushroom juice. And something I'm going to talk about in a moment is that mushrooms have, or fungi rather, have some of the most chemical complexity of anything in the natural world. They make compounds the likes of which nothing else even approaches. Incredible complexity. The medicinal value of the things that are in all sorts of different fungus is really starting to be explored now. People are isolating those compounds and figuring out what to do with them. But a lot of this comes from they're just extremely versatile creatures. So something I was mentioning a little while ago was that so that up there started as a block of oyster mushrooms that was growing on straw. And so they let it make mushrooms as many times as it was going to make mushrooms until it's what's called spent. So it's used up all the food. And then that guy took transmission fluid and dumped it on there, and they ate it and made more mushrooms. So they are phenomenal at breaking things down, be it crude oil. I've got a species that'll eat plastic. There's just all the best breaking down enzymes come from mushrooms, because that's what they do. That is their main job. On that topic, on the topic of sessile animals, they hunt. So over here, this is a nematode, which is a teeny tiny worm. That little lasso that it's stuck in is the lassos that oyster mushrooms have. So it'll wait until a nematode swims into the little thing, and then it goes shink, and constricts on it, catches it, and then breaks it down and eats it. So when you've got these things living in your soil, this is one of the ways that you help protect the ecosystem from nematode overgrowth, which can destroy plants and all kinds of stuff, and make things sick. But when you've got a fungus that can straight up hunt them, it's one of the things that help keeps everything in balance. Also, anytime that you see mushrooms growing on a log, that whole log is colonized. The mushrooms are just, as I said before, they're just the reproductive bit. So by the time that happens, they've eaten that whole log, basically. So it's just a matter of time before they'll turn the whole thing to dirt. Yeah, so this is what I was talking about. Immense chemical complexity. And something that I'm going to talk about in a moment is they tend to break down, as they're breaking down things like lignin, it actually generates a bunch of really interesting chemical species that are really convenient to then shove back into their own weird configurations to do mushroom things instead of plant things. So some of them glow. So I've had this species before. I'm trying to get my hands on it again. It really does glow quite nicely. You gotta be real in the dark for a while to see it, but they really glow. And so people have been trying to figure out how they do this. And I find this absolutely fascinating, that one of the compounds that is a result of breaking down lignin almost perfectly fits into this pathway that ends with this glowing molecule. So lots and lots of different fungi make that molecule, but not all of them have the enzyme that uses it as fuel to glow. So some of them, for whatever reason, have decided to pick this up. The running hypothesis as to why they do this is to attract creatures at night. So that way they go, ooh, glowy thing, and then they like rub up against it and get some spores on them and then run away. Because neither of them are particularly edible. Like you're not gonna want to eat either of these. But they're neat looking, so you'd probably be attracted to them if there's no other light. Excuse me. But there's this thing that kind of exists in the realm of like myths, which is foxfire, or like spirit lights. Basically, it was this idea that like if you went out in the woods, sometimes you'd see little glowy things, and it was the spirits. It was these guys. So like these are literally the foxfire mushrooms. So if you were, before we had flashlights and stuff, if you're in the middle of the forest, it's totally dark enough that you'd be able to see this, and your whole path would be lit up by all this glowing stuff. It'd be enough to like really go, oh wow, that's a thing. But yeah, it turns out it was just these mushrooms. So people have basically figured out their glowing pathway, and soon, hopefully, they'll be publishing that paper. Because once you know exactly how they glow, you can take those genes and put them into something else, which is something I want to do because it's so cool. I'd love a tank of that, right? I already had one, like a tube of this as a nightlight. Like, that'd be amazing. But also, you can make really interesting things. Because the nice thing is that the compounds that let these glow are fairly innocuous. Or at the very least, they're not gonna like freak your cells out. So if you wanted to make like a glowing fish, it'd be fine. Whereas if you tried to use the bacterial glowing pathway, some of the compounds in that pathway mimic molecules that are only present inside your mitochondria, which are the energy production parts of your cell, which mitochondria are technically bacteria. So if those compounds end up outside of the mitochondria, your body interprets that as mitochondria are exploding, therefore self-destruct because something has clearly gone terribly wrong. Whereas with this, that wouldn't happen because it is much more innocuous, in theory. But yeah, so now the actual like growing fungi bit. We did the first steps, but there's 3 main steps, which we did the first one, which is making the food, because they've got to eat something, right? As I was mentioning before, you can do hardwood pellets, you can do different kinds of flour, you can do just straight up birdseed. The nice thing with birdseed or like rye berries is because the shell is still on them, it acts as the like separation without having to add vermiculite. So it's both the food and the separator, makes it airy without getting too clumpy. But the main characteristics you're looking for are things that aren't going to clump too bad. Once you've done that, then you actually inoculate them, and so you either put in some spores or some growing mycelium, or just, there's all sorts of different things that you can use as your source of fungi. Some people literally just like carve a chunk out of a piece of mushroom and just put it in there, and the mushroom cells will be like, oh look, perfect growing conditions, and it like devolves from mushroom and turns back into mycelium and just grows. So then you just let it grow. You basically want to wait until the entire thing is white. So you can see these ones still have time, like they need more time to grow. But you go until once the whole thing is covered, then you're good. Because basically by the time the outside is covered, the inside is covered. So wait for it to grow, and then when it's fully colonized, then it's actually time to fruit them, which is making the mushrooms, which in our case is going to be really, really simple. All you do, take off the lid. You can actually put like a Ziploc bag on top, leave a whole bunch of headspace. The thing that you want, because you want to simulate the raining thing, so you spritz them with a little bit of like clean water, and then a lot of fresh air. So you like at least twice a day you want to give them a bunch of fresh air, maybe a little mist of water if they're looking kind of dry, and then put the Ziploc bag on top, and then they'll continue to do the thing. There's sort of a sweet spot. If you let them go too long, they're going to get gross. You basically want them when... So using these mushrooms, which we're not growing these ones. But yes, you basically want it where the cap has unfurled, but you don't want to go... If you keep going, the cap will tip all the way up, and then it releases its spores. Don't let it go that far. They're not good for you. I mean, they're fine to eat at that point. They're not as good, and they get kind of soggy. So there's a sweet spot where you want to just let them go just enough that they're as big as they're going to get, but not so big that they get all crinkly. But that's basically it. You just keep an eye on them. While they're growing like this, keep them in the dark. When it's time to fruit them, they need light. So put them not in direct sunlight, although we're in Canada, so does anything count as direct sunlight anymore? But yeah, no direct sunlight, but some amount of light, and then that fresh air, a little bit of humidity, a little bit of spritz of water, and they should just fruit no problem. If instead of doing that, you want to use the jar that you make to make more fungi, don't do that. You're going to have to basically make more food, get another jar, or a bag, or there's all sorts of different things you can grow in. We're using jars because it's convenient, especially for when you're just getting started. But some people will use like big bags. Some people use like laundry hampers filled with like sterile straw. But you can like cut a little piece out of this, of the mycelium, making sure that you do this in as sterile of a way as possible, and just transfer it to new sterile media, and it'll just take over and grow. So generally what people will do is they'll go from like a jar to maybe like a big thing of straw, and then from that they'll go to like a huge thing of straw, inoculate all of it, and then just grow a ton. So it really depends. If you just want to do this because it's just kind of fun, then just take the top off, let them fruit. If you want to propagate it, and then you can leave the top off and let them fruit. Some people will actually build like a whole fruiting chamber. So instead of just taking the top off, they'll build a special box. They'll line it with perlite, which is similar to vermiculite, but holds water a little better. And then you kind of put a raft on it, and then they take the whole jar and they cap it out so the whole cake comes out. And they just put the whole cake, and then the mushrooms will grow out from all sides. That is a little more efficient, but for the size of the jar that we're using, it's not necessary. It's easier to just take the top off, and frankly I've had better results just taking the top off than trying to dislodge the whole thing. Because when you do, then you gotta like wash them and let them grow more, and it's a nightmare. Just take the top off, it's way easier, especially at this scale. Okay, yeah, so the things that you can use to inoculate. I'm only showing two things here. There is a third option, technically. The main one we're going to be using are spore syringes, which is literally like they've taken some sugar water and they've mixed a bunch of spores into it and put it into a syringe. So all we're going to do is once the things have cooled, and they really do need to be cool or we're going to cook the things and that's no good, is we're going to take the syringe, light the end on fire, just briefly to sterilize it, and then jab it through the little red port and just aim it at the edge of the glass, like get it all as far in as it's going to go, but then like tip it so it's pointed towards the glass, and just a little squirt. You really don't need a lot. A couple of spores or a little bit of mycelium will do. Those spore syringes are heavy loaded, they're ready to go, you really don't need a lot. So do a little squirt and then take it out, light it on fire again, and then go into the other port, a little squirt on the other side. Technically that's overkill, but just to make sure that it works. Keeping in mind we only have 2 spore syringes and I need a little bit of the stuff left over so that I can make more for next time. The other thing that you can get, so spore syringes are great if you're doing jars. The other way that you can grow mushrooms is on logs. So what you do is you get a log that hasn't been, has freshly been cut down kind of thing, not something that has mushrooms growing on it already. And then you drill a bunch of holes in it, and you get these, which are wooden dowels that basically they've just filled a jar with those dowels and then squirted some spores onto it and just let it grow for a while. But then what you can do with those dowels is you kind of take them, drill a hole, knock them into the log, and then seal it with wax. And then that's a way of basically choosing which mushrooms get into the log. So you do this all over the log, and then in 6 months kind of thing, the logs just start putting out mushrooms like crazy. But we have some plugs. The way we're going to do it is we're going to open the jar. We're going to do this in the biolab so that the air is still and less dirty. We're going to wear gloves so that way we don't contaminate everything. And we're going to open the jar, drop a plug in, and then close the jar again. So those are our 2 options for today. The third option is you actually grow it on like a petri dish. So you can basically take one of those spore syringes, squirt a little bit onto the petri dish, let it grow, and then just carve a little chunk out of that and then drop it into your thing. So the nice thing with that is you can basically see how the individual spores are growing, and so you can pick one that's like, ooh, that one's really aggressive, let's take from that area. Or like, that one's really like ropey, for whatever reason, that's a characteristic that people seem to want in their fungi, because it means they tend to grow really, really well. So you'd be like, okay, I'm going to take a little ropey bit and like throw that in. Yeah, so the last note really is the type of mushroom you're growing decides what you grow it on. Most things that you can buy at like Myco Boutique are very forgiving. They're things that'll grow on pretty much anything. There are some exceptions though. Shiitake, for example, hate growing in jars. Absolutely despise it for whatever reason. They're a log kind of mushroom. On the flip side, on the right over here is Cordyceps. So these are actually a type of fungus that normally only grow on insects. So they infect an insect, zombify it, and take over its brain, drive it around for a little while, walk it up to the top, like the highest point it can find, and then immobilizes them, and a thing grows out of their head, and then releases spores. Extremely gruesome. However, they also have like really significant like medicinal use. So this particular species is Cordyceps Militaries. These are special because you can actually grow these in jars. You need special media, so you can't just use brown rice flour, but you can totally grow these in jars. Whereas the normal kind that people are used to is a different Cordyceps species that is literally worth more than their weight in gold because of how rare they are. So if you can find one of those and sell it before it goes bad, like it is literally worth hundreds and hundreds and hundreds of dollars. Because people love it for like traditional Chinese medicine uses a lot of different fungi, and that's one of them. And if you can get that, that's like the fancy. The reishi that we have and are going to be growing are actually another that are used in traditional Chinese medicine. I believe that it was like one of the translations of it's like the mushroom of kings, basically, or like the idea was the only people who normally got this were like kings, and they used it to like live forever, kind of thing. There's actually a fair number of studies that there's really interesting compounds in them, and it's quite good for you. I found out the hard way that it'll also interfere with your medication, so they are quite potent. that was a fun day. But yeah, so we've already done this bit. The next bit is the inoculation, which I already talked about. These are the instructions. But beyond that, I mean, there's really not much more to it. Fungi are a pretty relaxed thing. It's a lot of like you do a thing and then you wait, and then you do a thing and then you wait some more. So it could take. I mean, at the size of these jars, it'll probably only take maybe 2 weeks total for them to be fully colonized before they're ready to fruit. But bigger jars can take longer. So if you're doing like a big bag of something, it can take, a month to be fully colonized. If you're doing logs, it can take 6. Some of them are even weirder. So there's like kits you can buy to grow morel mushrooms, because you can't really grow them in the same way that you grow these. They're more of a, you put the spores down and hope they grow kind of thing. So there's ways of like prepping your yard. And then you put the spores down, and you're like, look, I built my special little box, and they grow over there. They're like, well, we've taken over your lawn, to hell with your box. But yeah, I mean, other than that, it's really a straightforward process. And so, again, we're going to just be injecting through the ports or putting in a plug, and that's basically it. I think that's all the... Oh yeah, okay, so then this is the at-home activity, which is fruiting them, which I've already talked about. But yeah, it's really as simple as just take a lid off. And that is it. That is all I have to say about fungi. Do you have any questions?
So, we can eat them, right?
Yes, yes. All of the species that we're going to be working with today are edible. Yeah.
When you fruit them,
Mhm.
so when you grab all the edible parts, and it will continue producing more edible parts as long as there's consumable.
In theory, yes. These jars are kind of small, so I'll be surprised if they'll do a second flush.
Okay.
But you can probably get 2 flushes. It kind of depends. Sometimes they cooperate, sometimes they don't. We've had blocks that just like kept making more mushrooms, and we're like, when is it gonna stop? But But sometimes, they're just like, nope, we're not making any. It's like, what? But I did the thing. It's sort of the way biology goes. Sometimes it doesn't play nice with what you want it to do, because its goals and your goals are diametrically opposed. But yes, in theory, it should make more. And the nice thing is you can actually take so what people who grow lots and lots of these will do is they'll end up just collecting all of their spent blocks, because they'll grow like huge blocks of the stuff. And then they'll just like bury it in their yard and wait. And you'll end up with like a mound that's like covered in grass and just mushrooms growing everywhere.
Uh-oh. He wants to do that now.
Well, so this is what we did. So I was living in Florida, and the house that we were renting was basically barren. Like, it was sand. There was no yard, it was just sand. So what we did was we contacted the local utility company. Whenever they had to cut down a tree because it was in the way of like power lines, they would just give the mulch away for free. So like they'd throw it at a wood chipper and then drive the wood chips to whoever wants them. So we put in an order of like, I'd like some free wood chips. And so we covered our entire lawn, or like our entire yard, in a foot of wood chips. Well, because it compresses and it breaks down. Because when you're going from sand, you need as much carbon and nitrogen as you can get on there. So we did that, and then we had all these different kinds of mushrooms growing, and so whenever a block was spent, we'd like break it into pieces and just bury it all over the yard. And then sure enough, we were like, oh look, some mushrooms! And like there was like a big old lion's mane just growing in our yard one day. And we could like, if we turned over the mulch, it was just white. It was just completely white with mycelium. So we basically terraformed that entire yard. So now there's like plants, and it's lush, and there's like stuff growing everywhere. And it's all just because, we had to reset the microbes, give it some carbon that the sun can't burn. But yeah, they're pretty chill organisms as these things go. Anything else? Any other questions? Comments?
I want to eat mushrooms now.
Yeah, this is sort of the nice thing with growing your own mushrooms. Like, the stuff that you can buy in the store, they're always like portobellos, basically. Yeah. Like, portobello mushrooms and the little ones, they're the same mushroom. You just let one grow longer.
Yeah.
Or it's like a different strain where like one of them just gets bigger, but it's the same mushroom, so they taste the same. Whereas all the stuff that we're going to be growing is very different. If you go to Myco Boutique and they're not out of the other species, lion's mane are my personal favorite. They look kind of weird. They don't look like your traditional mushroom. They look like this mass of tentacles, basically. But they're really, really good. They taste like lobster, which is really fun, and they also have kind of that like lobstery texture, which is really interesting. But also, if you eat them raw, like if you like dry them, powder them, and take them in like pills or whatever, there's compounds in that mushroom that trigger nerve growth, which is kind of neat. So it's like a nootropic, but it's a mushroom, which is just neat. And yeah, and there's a lot I had to kind of leave out about mushrooms just because it's a big topic. It's like a whole chunk of the tree of life. So there's a lot to be said. I mean, one of the other they do things like the nuclei, which is like where all the DNA is. They don't really differentiate their cells so much, insofar as it's kind of like long tubes of just nuclei floating around. So it'll just be like dozens of nuclei just flowing through this network. So they'll do really weird things where like, if they encounter something they can't eat, the nuclei will kind of like cluster there and sort of figure out how to chew on that thing. And once they've figured it out, it just redistributes that DNA throughout the whole rest of the thing. And they're like, oh look, we can eat this now. Or it's like they'll just like the nuclei will flow around to like as like information nuggets. It's very strange, very, very strange creatures.
After the lecture, once the jars had been removed from the autoclave and cooled, it was time to inoculate them. Using a spore syringe, we could inject a small amount of liquid into the sterile media through the gasket ports, making sure to aim the needle towards the side of the jar. And that's all there is to it. Everyone got to go home with a collection of different fungi to grow. In this case, pearl oyster, phoenix oyster, and reishi, but my collection is constantly growing. I hope you enjoyed this lecture and workshop-style video. If you did, please leave a comment and subscribe to see more. And stay tuned for announcements of the next workshops. That's all for now, and I'll see you next time.